In the first lecture, we've discussed what polygenic scores are and what limits their performance. Now we turn to a question that how do we measure whether a polygenic score is doing a good job? So in this video, we will cover different evaluation metrics for quantitative and binary traits, evaluation and visualization of prediction performance, and common pitfalls that can seriously inflate the results if we are not careful. Let's start with quantitative traits such as height, BMI, or blood pressure. The most common evaluation metric is the squared correlation between the observed phenotype and the polygenic score in an independent validation sample. This is the prediction R-square. Importantly, this prediction R-square is bounded above by SNP-based habitability. You can never explain more variance than the SNPs capture. In practice, we adjusted for the covariates like sex, age, and ancestry principal components, and we report the incremental R-squares. That's the R-square obtained from the full model, including both covariates and PGS, subtract the R-square from the non-model, where we only include the covariates. This slide shows what different R-square value look like. When R-square equal to 0, the PGS carries no predictive information. At R-square equal to 0.5, there is a strong linear relationship. At R-square equal to 0.9, the predictions are linearly perfect. Visualization could also help identify outliers in the real data analysis, so it's always a good thing to do. In addition to accuracy, we also care about bias because we don't want to systematically underestimate or overestimate the genetic values. If we regress the phenotype on the PGS in the validation sample, the expected slope is equal to 1 if the PGS is unbiased. This means that 1 unit increase in the PGS would lead to 1 unit increase in the phenotype. If the slope is greater than 1, then that means the PGS is downward biased. And if the slope is smaller than 1, that means the PGS are upward biased. Here are the examples of the unbiased, the upward biased, and downward biased prediction. They all have the same prediction R-squares, but they have different slopes. For diseases and other binary traits, evaluation becomes more complicated. The phenotype is no longer continuous, so linear model R-square does not behave the same way. While usually we found the cases generally have a higher PRS than the controls on average, the distribution can overlap substantially. So we need different statistics to quantify the prediction performance. These are the commonly used statistics for binary traits or diseases. First one is the pseudo R-square from logistic regression. This AUC or area under the ROC curve is also commonly used in the field. The best statistic is probably the variance explained on the liability scale, and we will talk a little bit more about this soon. And you can also evaluate the performance and visualize the data using the risk stratification and decimal odds ratio. Different statistic captures the different aspect of the prediction performance, and some statistic tend to be has a more appealing property than the others. We'll first look at the pseudo R-square. This is when you are using the logistic regression to calculate the prediction R-squares. So similar to the linear model, we have the now model where only includes the covariates, and the full model includes both covariates and PGS. Then we can calculate the pseudo R-squares. There are many pseudo R-square statistics available for logistic regression. Naga-Kirk R-square is one of the most commonly used one, which rescales this likelihood ratio to between 0 and 1. However, these values do not have the same interpretation as the R-square from the linear model, and this leads to important problems. So one of the major problem with the pseudo R-squares is that it depends on the proportion of cases in the sample. As shown in this graph, different colors shows the disease with different population prevalence. With the same PGS, but different proportion of cases in the target sample, we actually get a different pseudo R-square values. So that means the pseudo R-square is not portable across studies with different sample prevalence, and their results should be interpreted with caution. Another widely used metric is the AUC, or area under the ROC curve. AUC can be interpreted as the probability that a randomly selected case has a higher test score than a randomly selected control. So basically, it's looking at the sensitivity versus the positive rate. And we want the AUC value as high as possible. The AUC value of zero point 0.5 usually means there is no predictive value. There's no difference than a random guess because you are place the case over control at a probability of 0.5. And AUC value of 1 means the perfect classification, means you classify all of the cases above the controls. You can work out the AUC by this process. So you start with ranking individuals based on the PGS from highest to the lowest score. And you start at the region of the graph, work through the list of the ranked individuals. You can move 1 unit along y-axis if the next individual is a case, and move 1 unit along x-axis if the next individual is a control. And then once you have this ROC curve, you calculate what's the area under the ROC curve to get the AUC number. There is a nice property about the AUC statistic. The key advantage is that it is independent of the case control proportion, making it useful for comparisons across studies. However, the downside about that is the maximum value of AUC will depend on the disease heritability and population prevalence, as shown in this graph. So comparisons across populations with different disease prevalence would require caution. Compared to the previous two statistics, this prediction R-square on the liability scale is most recommended. To better interpret the results for disease, we often convert the prediction accuracy to the liability scale using a liability threshold model. So this shows the basic assumption of the liability threshold model that we have the liability that is the normally distributed, and the disease status reflect whether this unobserved liability exceeds the threshold. And this threshold is determined based on the population disease prevalence, K. So for most of the disease, we don't observe the liability underlying the disease. But you can think of one real life example, the BMI and the obesity. So BMI is like the observed continuous liability for obesity, where the individual get obesity only when is the BMI past some predefined threshold. And using this framework, we can map variance explained on the observed 0-1 scale to variance explained on the underlying liability scale. We can do that by first estimating the prediction R-square on the observed 0-1 scale using the regression models. And then we can map this observed scale R-square to the liability scale, depending on the disease prevalence K and the density of the normal distribution at the threshold Z. And the theory is illustrated in detailed paper in 2012. In the case control studies, samples are often ascertained, means that the sample prevalence different from the population prevalence. And in this case, the ascertainment need to be corrected when we are mapping to the liability scale. Um, and the reason we are doing that is once you do the ascertainment correction and map to the liability scale, then the variance explained by the predictor will be comparable to the heritability estimate from the family data. So this slide shows the formula to correction for the case control ascertainment. It's also explained by this Li et al. Paper. Although the formula looks complicated, the key message is simple. Correcting for ascertainment is essential for valid interpretation of disease risk prediction. So why liability scale R square is useful? Well, it has a nice property that the heritability is independent of the disease prevalence. And once we map this prediction R square to the liability scale, then we can directly compare it to the heritability estimate from the family studies or from using the genotypes. And this provides the direct measure how well the predictor performs relative to capturing all genetic variation. And this makes it one of the most interpretable metrics for disease prediction. Another common way to present the results is to look at the risk stratification. You can stratify the samples based on the percentile of the Pontian score and look at the proportion of cases in each of the percentile of PRS. For example, in this landmark paper, Kirin et al., they found that for psychiatric disorders, if you look at the top 1% of the individual in the PRS distribution, 1 in every 9 would actually the cases for the coronary artery disease. And this is equivalent to the risk conferred by the monogenic mutations that once detected, there is actionable clinical management can take place. So this is particularly relevant to identify the high-risk subgroups for disease prevention. Another common visualization is to divide individuals into deciles based on their PRS and compare disease odds ratio between groups. For example, you can calculate what's the odds ratio of the top decile to the bottom decile. And you can also calculate the top decile to the medium decile. This is a very good visualization. Um, um, and it's quite intuitive. But the metric is sample dependent and it's not easily compared across different studies. So this slide shows an example where when we look at these odds ratio in different cohorts, and this is what the results look like. If you compare the top decile to the bottom, this odds ratio can be as large as close to 25-fold. But these cohorts are case control samples. That means the number of cases are highly ascertained. If you scale these values to the population risk using the same data, the results actually looks much less impressive. And the relative disease risk could get down to up to 7.5%. Sorry, 7.5-fold. This slide shows the toy example illustrating how these odds ratios can be calculated from the simple case control counts. So we have the counts of the cases in the first and the bottom decile of the PRS, and you have this number for the controls. You can look at the odds being the cases in the 10th decile and the odds being the cases in the first decile, and you calculate the ratio of the odds. And that's how you get the odds ratio. You can also get this from the logistic regression. Now we come to the pitfalls. This is very important, and I've seen some papers published in the top journals that made these mistakes. So we had better to do it correctly in our own study. So for polygenic prediction, usually we have three samples. One is the GWAS discovery sample to train the data. So it's also called the training sample. Um, and if you're using some conventional methods that requires the tuning sample, then we have another sample to tune the parameters to build the best model. Sometimes it's also called the validation sample. And then we have the target sample we want to apply the polygenic risk score to. And usually in this sample, we don't have the observed phenotypes. And it's called, sometimes it's also called testing sample or validation sample. The fundamental requirement is that all of these samples need to be kept absolutely independent. Any overlap between these samples will lead to inflated prediction accuracy and invalid results. So one obvious pitfall is or mistake is that you report the prediction R square in the discovery sample itself. It's also called the in-sample prediction. And from some simple derivation, you can see that even under the null model, there is no association between the genotype and the phenotypes. Using the same data to train the prediction equations would lead to a chance variation. And this prediction R square under the null hypothesis is a function of the number of SNPs and the sample size. This is a classic overfitting problem and is also known as the winner's curse. So doing the out-of-sample prediction is essential. The second pitfall is that you are not doing the within sample prediction, but the target sample is partially overlapped with the discovery sample, and that could also inflate the prediction accuracy. A less obvious case is that although the target and discovery samples are different samples, but they have a greater similarity. For example, they are genetically related. And if the goal is to predict someone like a random person in the street using the training samples that has including the genetic relatives to the target sample would inflate the prediction accuracy. Just a note that it's common that people do the cross-validation within the total sample, leave out one fold, and as a validation and train the data in the rest of the data, and then rotate the holdout samples. This is not really the pitfall, but need to be careful if the different folds share the same confounding factors. That could also, to some extent, inflate the prediction accuracy. So doing a cross data sets or cross biobank prediction is the gold standard to test the prediction accuracy. The last pitfall is the least obvious of non-independence. That is, if you estimate the SNP effect or select the SNPs to go into the prediction equation using a total sample that include the discovery and target sample, and it could lead to inflation, even though you re-estimate the effects in the tuning sample. So as shown in this graph, the blue line shows if you do everything properly, this is what the prediction accuracy will look like for the out-of-sample prediction. The yellow line shows if you do the within-sample prediction, estimating the SNP effect in the same sample, the prediction accuracy literally goes to 1. The red one is if you select the SNPs within using the total sample and re-estimate its effect in the tuning sample, you still got the inflation. It's because the SNPs are estimated not only because of the true signals, but also associated with an error, and that error cause overfitting if the target sample was also included in the selection of the SNPs. Okay, so in summary, assessing prediction accuracy and bias is important and straightforward for quantitative traits. For disease traits, it gets little bit more complicated. We have different statistics to evaluate that. Serial R square is a popular one, but it's subjected to the sample prevalence. AUC is widely used, but the maximum value would depend on population prevalence. So comparing studies with different prevalence would require caution. R squares on the liability scale is the most appealing methods because it's independent of both population prevalence and the sample prevalence. And it's directly comparable to the heritability estimates from the family study. Nassau odds ratio is great for visualization, but it's not portable across samples. And most importantly, we need to pay attention to the pitfalls in the prediction analysis and absolutely make sure that the discovery and the target samples are completely independent. And these are the excellent reference for this lecture. Thank you.